Bismillahirrahmanirrahim. The synthesis of steroid hormones is discussed in this video. The steroid hormones are lipid-soluble compounds and they are chemically derivatives of cholesterol. Structurally, they consist of 3 cyclohexyl and 1 cyclopentyl ring, forming the core cyclopentanoperhydrophenanthine structure. The steroid hormones are not stored in large quantities as active hormone. Instead, cholesterol esters are stored in cytoplasmic vacuoles and serve as the reservoir for rapid hormone synthesis. The cholesterol is taken from the plasma as LDL particles, and there is also de novo synthesis inside the steroidogenic cells such as adrenal cortex, gonads, and placenta. The examples of these hormones are cortisol, aldosterone that are secreted from adrenal cortex, and progesterone, estrogen, and testosterone that are secreted by the gonads. Placenta secrete estrogen and progesterone during pregnancy. Their release mechanism are not packaged in secretory granules, and instead, upon synthesis, they are freely diffused across the lipid barrier of the cell membrane. They enter the interstitial fluid, then bloodstream, and bind to the carrier protein such as albumin, corticosteroid binding globulin. So their speed of action is slower than the peptide hormone, but they have got a longer duration. And the mechanism of their action typically is through the intracellular, both the nuclear or cytoplasmic receptors, and regulate the expression of the gene. Because they are lipid soluble and circulate uh, with the proteins, means they are bound to protein, steroid hormone levels are measured as total, bound as well as free. But free hormone is the biologically active form of the hormone. This image illustrates the chemical structure of 4 major steroid hormones, all derived from the cholesterol. Each structure retains the core of the cyclopentanoperhydrophenanthine ring that that is common to all steroid hormone in the aldosterone, cortisol, estradiol, and testosterone. And there are 3 6-member ring, while one 5-member carbon member ring. So the aldosterone is classified as mineralocorticoid and it is secreted by the zona glomerulosa of the adrenal cortex. Its function is to increase the sodium reabsorption and potassium excretion in the kidney, and also contribute to the blood pressure regulation. It possesses an aldehyde group, CHO, and hydroxyl group, include a ketone group and a formyl group, which are key to its mineralocorticoid activity. The estradiol is estrogen. It is produced by granulosa cells of the ovary It promotes development and maintenance of female reproductive structure and secondary sexual characteristic. It contains an aromatic airing, a defining feature of estrogen, has hydroxyl group at position 3 and 17, critical for estrogenic activity. Then there is cortisol, that is glucocorticoid. This is secreted by zona fasciculata of the adrenal cortex. It regulates metabolism, suppresses inflammation, and helps the body respond to stress. Its structure, major structure, it contains a hydroxyl group at position 11, 17, and 21. Has a ketone group at position 3 and aldehyde related side chain as C17. Testosterone is androgen. It is produced by Leydig cell in the testes. It promotes the development of the male reproductive tissue and secondary sexual characteristics. And its structure is that at 17 it has got a hydroxyl group, and at position 3 there is a ketone group, and it also retains the 4-ring core structure with double bond in the A-ring. These molecules differ in their function, group, and degree of saturation, that is aromaticity, which determine their biological specificity and receptor affinity. Despite having a common cholesterol backbone, these modifications create a wide range of physiologic effect. Thank you for watching this video. If you have any query, please write in the comment box.